Hi, my name is Harbinder Kaur. I am a PhD student at Jawaharlal Nehru University. Today I am going to talk about my work on benchmarking, taxonomic classification, metagenomic tools for virus variant and strain identification. Identifying microbial taxa present in a complex biological and environmental samples is an emerging challenge. Methods have been developed recently to identify microbial taxonomic group from a mixture of sequence reads. The redeployment of a similar framework for sequence-based virus taxonomy is necessary for the genomic surveillance of existing strains and to identify the novel strains. To benchmark widely used metagenomic classifiers like Kraken2, Centrifuge, Gotcha, Metaflen2 on virome data, we used 6 species level and 5 strain level simulated virome data as shown here in this schematic phylogenetic tree. Kraken2 uses a Kamer to lowest common ancestor mapping approach, whereas Centrifuge uses a indexing scheme based on Burrows-Wheeler transform and Ferenczi-Manzini index to create a compressed database for rapid classification. Gotcha is an alignment-based method, and Metaflen2 uses a database of unique clade marker sequences. Default databases were downloaded with all the 4 tools, and custom databases were also built for Kraken2 and Centrifuge from the FASTA files of these viruses. Usher is a strain level identification method, and it requires a pre-existing phylogenetic tree to place new sample onto. Species-specific trees were prepared for Usher. Here we can see Kraken2 and Centrifuge perform the rapid taxonomy identification as compared to other tools. Kraken2 default database is different for viruses and bacteria, etc., whereas Centrifuge database is a compressed database of bacteria, archaea, viruses, and human. So to perform the taxonomic identification with Kraken2, we only use the viral database, and it was faster as compared to Centrifuge. But Centrifuge was faster than Kraken2 when used with custom database. All the tools provided species level taxonomic identification but were not able to characterize the strain. To identify the strain level taxonomy, we used Centrifuge in tandem with Usher. From Centrifuge results, read IDs and taxonomy IDs were extracted. The taxonomy IDs were used to build species-specific phylogenetic tree for Usher. Usher requires input in form of a VCF file, so we used GATK pipeline to get the VCF file from the FASTQ reads. To build the mutation annotation tree for Usher, we used phylogenetic tree corresponding to the tax IDs extracted from the Centrifuge result and VCF file extracted from the FASTA sequences of the phylogenetic tree. Then it places the read VCF file onto the mutation annotation tree to classify the strain based on maximum parsimony approach. This slide shows the result of the proposed approach. Centrifuge identified the species from the FASTQ reads, and Usher further provided strain level taxonomy. Phylogenetic tree was built for SARS-CoV-2, and all the strain for variant of concerns were colored differently. The simulated reads from SARS-CoV-2 beta, delta, omicron were successfully placed on their respective clades.